acknowledge some of the colleagues and collaborators that I work with at the New York Botanical Garden, as well as, can you see this pointer? Or is that kind of useless? Yeah, it's there? Okay, where'd it go? There. These three students are actually high school interns that have helped us out in the summer towards this work, and NSF for funding. So I'm interested in floral development and the genetic basics behind how flowers are formed, especially in how differences in gene expression and function between species relates to the differences in phenotype that we see in flowers of different species. I'm also interested in the consequences of gene duplication and how duplications have affected floral evolution. So gene duplication can occur in several different ways. We could have whole genome duplication, where we actually get a duplication of the entire genome, so all of the genes that are present in a species. So going from something that's diploid and has 2 sets of chromosomes to something that's tetraploid and has 4 sets of chromosomes. You can also get gene duplication on a smaller scale, things like tandem duplications, where a single gene or block of genes on a chromosome can be duplicated, or things like single chromosome duplications. But all of these duplications mean you have 2 genes where you used to have only 1. And following duplication, we can get several different gene fates that occur. So we could get the maintenance of both copies. We have copy A and copy B, and they are redundant. They do the same thing. They do the same function that the original gene before it duplicated did, and so we have those 2 copies that are maintained. We could get gene loss, and that's where one copy acquires mutations and becomes non-functional, becomes a pseudogene, and is eventually deleted from the genome. So we're just left with, again, 1 gene where we originally had 1 gene. And then the more exciting things, I think, we can get neofunctionalization, where 1 gene copy goes on to acquire a new function, while the other copy maintains what we saw in our original gene. And we can get self-functionalization, and part of self-functionalization is partially tissue-specific expression, where, for example, 1 copy could be expressed in the leaves, and 1 copy could be expressed in the roots. But it's also about the partitioning of original gene function. So your original gene, before it was duplicated, had this set of functions, and now these 2 genes take, okay, gene A does some of the functions, and gene B does the other ones. But together, they do the same functions that the original gene used to. So these are some of the fates that we can expect after genes are duplicated. So I'm going to give you some background in floral anatomy and the genes that determine how flowers are formed. So here we have the cross-section through a flower. And you can see that we have 4 different organ types. So here, on the outside whorl, in the first whorl, we have sepals. In our second whorl, we have petals. In our third whorl, we have stamens, which are the male reproductive parts of the flower. And in the middle, in the fourth whorl, we have carpels, which are our female parts of the flower. And there are several different genes involved in determining which organ is produced. And the model behind this is called the ABC model of floral development. So here we have these 4 different classes of genes. We have A-class genes, B-class genes, C-class genes, and E-class genes. And their overlapping expression patterns determine which organs we get. So A-class genes are expressed in the first 2 whorls of the flower. So here we have a mature flower, and here we have a developing flower. So these little bumps are the developing organs, very early in development. And so A-class genes are expressed in the first and second whorls of the flower. B-class genes are expressed in the second and third whorls of the flower, and then C-class genes are expressed in the third and fourth. So if you have only A gene expression, then you get sepals. If you have A and B gene expression, then you get petals. If you have B and C, then you get stamens. And if you have only C, then you get carpels. So that's the basis of how we get these different floral organs. And interestingly, in all of these A-class, B-class, C-class, and E-class genes, we see that there are gene duplications in those gene lineages. So here we have have some phylogenies, and each of these trees is a gene tree. So based on molecular data, we can see which genes are more closely related, and from the branching topology, we can see whether there have been duplications that have occurred. And these triangles represent groups of closely related genes. And the stars that we see at these branches are where duplications have happened. So you can see that there are quite a lot of duplications in these genes that are involved in floral development. And I'm interested in one particular lineage of these genes, which is the apetalal-1 fruitful gene lineage. And you can see from all of these stars, there it is, you can see from all of these stars that there are several duplications that have happened in this gene lineage, but we're specifically interested in this yellow one here, which is the AP1U full duplication, which occurred at the base of the cordydicots. And cordydicots are a group of plants that are related to each other, and this duplication occurred about 60 million years ago and resulted in our UAP1 genes and our U full genes. And then we also have, before this duplication, here in these more early diverging lineages, we have these full-like genes, which were called full-like because they resemble the full gene more than they resemble the AP1 gene. So full genes and AP1 genes differ in the protein motifs that they have. So here we can see in full genes that we have this full-like motif, these 6 amino acids, and you can see that the full-like genes and also some related genes have the same full-like motif. But our AP1 genes have 2 new motifs, this farnesylation motif at the very end of the protein and this transcription activation domain. And it seems that these new domains in the UAP1 genes occurred via a frame shift in these proteins. So here we have the DNA sequences from 2 genes. This gene is a full-like gene, so again, one of those early diverging lineages that I showed you before. And then here we have a UAP1 gene. And transcripts have different reading frames. So 3 different reading frames, and a couple of them are shown here. We could have this reading frame, or we could have this one, which is slightly shifted. And you can see that these DNA sequences both show the potential to have this full-like motif that we see here in teal, or this farnesylation motif that we see in purple. And which one we actually get in the protein is determined by which frame the protein is reading at that point. And in our full-like gene, we get this reading frame, so we get the full-like motif. But in the AP1 genes, we get this reading frame, so we get the farnesylation motif instead. So it seems like this new farnesylation motif is due to a shift in the protein, in the reading frame of the protein. So we get these 2 different motifs. So AP1 and full have different functions in Arabidopsis thaliana, which is a model organism in the mustard family of plants. And AP1 and full are expressed in mutually exclusive domains. So here we have our AP1 expression domain, and here we have our full expression domain. And you can see that if you took these 2 photos and you overlaid them, that those expression patterns domains wouldn't overlap. So they're being expressed in different places within the developing flower. And both of these genes are required for floral meristem identity, and meristems are basically the stem cells of the plant. So they're small, undifferentiated, dividing cells which give rise to the organs of the rest of the plant. And the floral meristem is a meristem that is committed to becoming a flower. So both of these genes are involved in creating those floral meristems. But they also have some differing functions. So AP1 is involved in sepal and petal development. And remember I told you before that sepals and petals happen in the first and second whorl of our flowers. And if you knock out AP1 function, then in our AP1 mutants, sepals are replaced with bracts. So bracts are leaf-like organs that are present in the reproductive part of the plant. And so we get these bracts instead of sepals in our first whorl in AP1 mutants. No petals are formed in our AP1 mutants at all. And we also get these secondary flowers. So instead of having one flower, and here's our wild type flower. We only get one flower per pencil, so that's the little branch that each flower is on. But in AP1 mutants, we get lots of flowers. We have secondary flowers and sometimes tertiary flowers, and a whole lot of flowers. Whereas in full, that gene is involved in fruit development. And here we see a wild type fruit. It has these long, elongated fruits. And in full mutants, where there's no full function, we get short fruits that fail to elongate, and then there's a premature rupture of the fruit wall, like we can see here. So we know that these genes have different functions and that they occurred from a duplication, but is that functional divergence that we see between the two genes due to subfunctionalization, that partitioning of original gene function, or neofunctionalization? Has one of these gene clades gone off and done some new function? And in order to answer that question, we can look at the pre-duplication gene functions. Remember I talked about those full-like genes that diverged earlier in the tree? We can take a look at those genes and see how they function in order to determine whether AP1 full underwent sub or neofunctionalization. And some colleagues at the Garden did virus-induced gene silencing, which is we can use to knock down these genes in some pre-duplication genes from the poppy family. And here we have California poppy, and in this column we have our wild type, and then in this column we have our full-like mutants. So here we can see the branching pattern of what wild type, what we expect, and what's normally there. And then in our full-like mutants, you can see that there is more branching happening. Here we have a bud, a wild type bud, and you can see these nice smooth sepals. But in our mutant, you can see that they have a bract-like quality, like we were saying in the AP1 mutants in Arabidopsis. And you can see that there are lobes, which is similar to what we see in the leaves of wild type. So they're taking on a more leaf-like quality. And in wild type, the sepals actually fall off of the flowers when they are mature. But we can see that here in the mutants, that these leafy type grafts are still present on the flowers. Here we have another species within this poppy family, which is opium poppy. And here in the wild type, you can see the branching pattern. And then the full-like mutant, again, we see more branches, just like we did in the other species. Here we have our wild type flower with nice white flower petals. And in our mutant flower, we can see these green pieces. So we're losing some petal identity. And here's our wild type fruit. And again, we get this ruptured fruit in our full-like mutants, like we saw in the full mutants in Arabidopsis. So if we put all of this together, here we have our gene tree again. So these purple branches are AP1 genes, and those are genes from different species that still have that AP1 identity. Here are our full genes, again from different species. Here are the full-like genes that I just told you about in the poppy family. And across the top here, we have the different functions that these genes are doing. But what I want you to take away from this slide is that if we look at our pre-duplication full-like genes, you can see that these genes are doing all of these functions. But if we look at our AP1 and full genes, then AP1 is doing this one, and full is doing these ones and this one. So it looks like AP1 and full have partitioned the function of the original gene. So it looks like we've gotten subfunctionalization in AP1 and full based on the functions that we saw in our pre-duplication genes. So that was all background. Now let's get into what I'm actually doing in my research. So these are my questions. So we know that AP1 and full have different functions. We think it's due to subfunctionalization since the duplication, but how has that functional divergence actually occurred? Is it due to changes in the regulation, so where genes are turned on or turned off? Because like I said before, we know that AP1 and full are expressed in different places in our developing flower. Or is it due to changes in the protein coding sequence? We talked about those different protein motifs that AP1 and full have. Or is it a bit of both regulation and changes in our protein coding sequence? And to answer this question, we can do promoter swap experiments. So genes have two parts. They have a promoter, which is the regulatory part, so where the genes are turned on or turned off. And then the second part is the coding sequence, which actually encodes for the protein that's formed. So if we take the promoter of AP1 here and put it with a coding sequence of full, we can actually express full in the AP1 domain. So here's our AP1 domain, and that's where AP1 is usually expressed in wild-type plants. But we're going to make it so that full is expressed there in order to see whether full can substitute for AP1 function. So can full do what AP1 normally does? And so we're going to transform AP1 mutants, which don't have any AP1 function, with this construct where we're putting full in the AP1 domain to see whether full can rescue those AP1 mutants to look like wild type, so look like what we see in a normal plant. And then vice versa as well, where we're taking the full promoter and putting it with the AP1 coding sequence, which will then express express AP1 in our full expression domain to see whether that construct can rescue full mutants to look like wild type. So those are our two experimental constructs, these ones here, but then we also have two positive controls. So those are on the bottom. So our AP1 construct, if we put an AP1 construct into AP1 mutants, then we should be pretty sure that it will rescue the mutants to look like wild type again. Because we're basically giving the mutant, which doesn't have any AP1 function, the ability to have AP1 function again, because we're putting that AP1 back into the AP1 domain that works. So we should get full complementation to look like wild type. And similarly with our full positive control into full mutants. So that's one question. And then we have a second question of what role do these conserved motifs actually play? So I talked about the full-like motif and the farnesylation motif. What are they doing? Are they doing something that's really important for protein function? And so to answer this question, we've made mutant coding sequences, and then we will transform those mutant mutant sequences into mutants to see whether they can rescue them back to wild type. And so we have our AP1 mutant AP1 construct, and these letters are amino acids, so part of the protein. This farnesylation motif, this cysteine here, the C at the beginning, actually accepts the farnesyl group for post-translational stuff onto the AP1 protein. So what we've done is we've mutated that cysteine to a serine, so it can't accept that farnesyl anymore. And so we're seeing how that will affect protein function. And then we'll put this mutant construct into AP1 mutants. And then we have three different full mutant proteins. Our first one is pretty drastic. So this full-like motif is near the end of the protein. And so we created a protein which actually truncates the protein right before this motif. So just ends right there to see how this motif helps to function in full function. And then we have some finer mutants where we've actually mutated some of the amino acids in this full-like motif. And I want to draw your attention. So this tryptophan, this W here, we see this tryptophan in this place in all of the full-like and related proteins that we've seen. So it's strictly conserved, so we see it all the time. So we think it might be doing something important. So we've mutated it to see how that affects the gene function. So let's move on to our AP1 promoter experiments. And these experiments will actually answer about half of the questions from each question 1 and question 2 that I just told you. So our AP1 full experimental construct will answer half of the question of our first question of our promoter swap experiments. So can full substitute for AP1 function? And then this our AP1 mutant construct will answer half of our second question on how these protein motifs are actually affecting the gene function. So can the mutant AP1 protein substitute for the AP1 function? And that seems kind of crazy. Why are we putting two different questions into the same experiment? But it's because they share the same controls. So here we have our AP1, AP1 positive control, and here we have our negative empty vector control. And we need both of those controls for both of these experimental constructs, so we put them together in one big experiment to see what happens. And so I transformed each of these constructs into AP1 mutants, and this is what we get. So in our AP1, AP1 positive control, we can see that we have petals, unlike in our AP1 mutant, which is really good because it's always good when your positive control works. That's a good thing. In our AP1 full lines, we can see that we don't have any petals. And in our AP1 mutant AP1 lines, we do have petals. And in our negative control with our empty vector, we don't have any petals, and it looks very similar to our AP1 mutant. So let's look at this a little bit closer. So here is our wild type planets for reference. And here we have two lines of our positive control, our AP1, AP1 constructs. This one you can see has petals, but if you look at these first world organs, they're actually still bract-like. Remember we talked about in the AP1 mutants we have bracts instead of petals in the first world organs. So it's not entirely rescued to the wild type, but this line looks much more similar to the wild type because it has petals and sepals. And here we have two of our AP1 mutant AP1 lines. And again, we can see they have petals and sepals and look very much like wild type. So here we have some expression of this AP1 construct in these different lines. So here we have wild type, so that's what we expect. And here we have our AP1 mutant. So you might ask me, well, why are we seeing AP1 expression bands in our AP1 mutant? And that's because even though AP1 is non-functional in these mutants, it still expresses an AP1 transcript. And actually, the transcript has the third intron left in the transcript instead of being taken out. And that's why it's non-functional. And you can see that the bands in the AP1 mutant are higher than the ones in wild type, and that's because it still has that intron in the transcript. And if we look at our AP1, AP1, and AP1 mutant AP1 lines, you can see that there are two bands. And that's because those are in AP1 mutant background. So they were AP1 mutants first, and then we transformed them with our experimental constructs. So they're going to have that AP1 non-functional transcript, but then they also have the wild type bands that we put into them. And I want you to see that in this first AP1 line, it has a lower expression in this wild type sized band than in the other ones that we see. And that correlates to the fact that this one, the phenotype didn't rescue quite as well. So it might be the fact that we don't see sepals in this line may be partially due to the fact that AP1 wasn't quite expressed at a high enough level to make sepals happen. So now we move on to our AP1 full lines. And again, here we have wild type and our AP1 mutants for comparison. And here we have three AP1 full lines. And you can see that there aren't any petals, like wild type, but they do seem to have fewer flowers per pedicel than our AP1 mutants. So this picture is actually from what we would expect a single flower to be in wild type. So it has multiple flowers where we expect only one. But, and here this illustrates it even better, here we have the first flower from an AP1 mutant, and you can see that there are lots of flowers going on here. We have primary flowers, secondary flowers, tertiary flowers, and I think there are even some quaternary level flowers in here. So lots of flowers where we expect only one. And then in our AP1 full lines, we can see that we still have multiple flowers where we expect one, but it's much fewer than we're seeing in our AP1 mutant. So taking all of this flowers per pedicel data, so here we have our different treatments in the different bars, and then the number of flowers per pedicel. And you can see that in our wild type, AP1, AP1 positive control, and our AP1 mutant constructs, we have one flower per pedicel, just like we expected wild type. In our AP1 mutants and in our negative control empty vector, we see quite a lot of flowers. And you can see that our error bars here are pretty big, and that's because it's pretty variable how many flowers are actually there. But way more flowers in these two than we see in our wild type. And if we look at our AP1 full lines, we can see that they're not quite down to 1 like we see in wild type in our AP1 positive control, but they're much fewer than what we're seeing in our AP1 mutants and our negative control. So here are flowers per pedicel in all of our experimental constructs. So now let's move on to the number of petals per flower. So here we have our primary flowers, and in wild type we have 4 petals, and in our AP1 positive control we also have 4 petals, pretty much. In our AP1 mutant AP1 lines we also have 4 petals. But in our AP1 mutants and our empty vectors we have no petals at all, and we have pretty much no petals in our AP1 full lines. So full is not rescuing AP1 to make these petals. If we look at our secondary flowers, well, why do we only have a couple things here? That's only because we only see the secondary flowers in the AP1 mutants and our AP1 full coding sequence lines. So in our AP1 mutants, we have no petals in these secondary flowers, and in our AP1 fulls, we can see that there are some petals sometimes. And you might say that the difference between 0 and half a petal is not very big, but it is in fact significant. So we're seeing significantly more petals in our AP1 full lines in our secondary flowers than we do see in our AP1 mutants. So a little bit more background. I'm going to talk about first world organs. Remember, those are the ones on the outside of the flowers. But there are different positions in these flowers, and I just want to give you an overview. So based on the main stems, and that's what we see here, we have adaxial first world organs, which are towards the main stem. That's those ones. We have the lateral ones, which were in the middle, and those are kind of perpendicular to the adaxial and abaxial ones. And then our abaxial ones are away from the stem. So that's just the position, and I'm going to talk about those in different categories. So here we have our first world adaxial organs. And again, we have our wild type and our AP1 mutant for comparison. So these graphs basically show we have all of these different categories of organs that we see in these treatments. And this is just a percentage of how much of those organs we see for each of these treatments. So if we look at the wild type one, you can see that it's all purple, and that means that it's 100% sepals. So that's what we expect in wild type, and that's what we see. But in our AP1 mutant over here, you can see this really big blue bar, and that's none. So those organs are just missing. They're not there. They're either aborted or just they're not there at all, and so we can't score them as anything. And then the other ones seem to be different sorts of bracts, those leaf-like organs that we see. And if we look at our AP1 positive control, you can see that we have quite a few sepals, this purple bar, like wild type, but then we have this greenish type bar, and that's sepals with Y-shaped trichomes. And trichomes are hairs that we see on plant organs, and the Y-shaped trichomes are a marker for leaf-like organs. So we see them on leaves, we see them on bracts, and we don't usually see them on sepals. But I'm seeing these sepals that have Y-shaped trichomes. So I wanted to put those into 2 different categories because they're different. But they're still sepals, so pretty much we see that our AP1 positive control is very much like wild type. So if we move on to our AP1 full lines, we can see that this blue bar here is much smaller in the AP1 full lines than in our AP1 mutant, and that means that we're missing much fewer of the first world adaxial organs in our AP1 full lines. So there are more organs there than in the AP1 mutants, and again, those seem to be different kinds of bracts. So we're looking at the same thing in those lateral perpendicular organs. Again, in wild type, we see mainly sepals. In our AP1 mutants, about half of the organs are missing, but the other half are different sorts of bracts. In our AP1 positive control, we see again sepals, and those sepals with the Y-shaped trichomes, again very similar to wild type. And in our AP1 full lines, here this blue bar of missing organs is higher than in our AP1 mutant. But again, different sorts of bracts are actually seen. And the last one are the first world organs, the abaxial ones. Again, in wild type, we have sepals. In our AP1 mutants, we're missing about half of the first world abaxial organs, and the others are bracts. In our AP1 positive control, we see sepals and sepals with Y-shaped trichomes, which look similar to wild type. And in our AP1 full lines, you can see that very few of the organs are actually missing. So we have most of our first world abaxial organs present, and those ones seem to be different sorts of bracts again. So now we're moving on to our second world organs. So these are where we expect petals to be, and that's what we see in wild type. Here we have 100% petals. And then in our AP1 mutants, again, about half of the organs are missing, but then we have all of these different organs. And remember at the beginning I talked about sepals, petals, stamens, and carpels were the 4 organs that we see in flowers. But if you look at this list of things, we see things like carpeloid stamen and petaloid stamen and carpeloid bract. So basically we're getting a mashup of the different organ types into one organ, which is kind of interesting. And if we look at our AP1 positive control line, you can see that it's all petals just like wild type. So that's doing really well at rescuing. And then in our AP1 full lines, we can see that there are fewer missing organs, but relatively the same things that we're seeing in the AP1 mutants, except that we have 2 new categories that we see in our AP1 full lines that we don't see at all in our AP1 mutants, and that is petals. So we see those few petals, and they're not quite like the wild type petals. They're a little bit green and they're a little bit folded, but they're still petals, so I put them in. And we also see these petaloid stamens, so stamens that have some petaloid characters, which we don't see in our AP1 mutants. So those were the primary flowers, and now we're going to move on to the secondary flowers. So here we put them all into the same graph, so I have our adaxial, lateral, and abaxial positions of these first world organs, and here we're comparing our AP1 mutants with our AP1 full lines. And you can see that in each of our AP1 full lines, that we have this blue missing organ bar is shorter than in our AP1 mutant. So we have more first world organs in our AP1 full lines. And also, in our adaxial and abaxial positions, we're seeing more unfused carpels in the AP1 full lines than we're seeing in our AP1 mutants. And so remember, the carpels usually are in the 4th row, in the middle of the flower, and we're seeing them actually on the first row, on the outsides. So that's kind of interesting. And then looking at the second world organs in our secondary flowers, you can see that the missing organs is about the same between AP1 mutants and our AP1 full lines, but we can see that petals are much more prevalent, and petaloid stamens as well in our AP1 full lines than in our AP1 mutants. So that was a lot of data. Good job, you got through it. So just to summarize, what are our questions again? So can full substitute for AP1 function? Well, yes and no. So no, because we don't see any petals in our AP1 full lines like we do in wild type. But yes, because we see fewer flowers per pedicel, and because there seems to be an increase in the number of the first and second world organs that we see in our AP1 full lines in comparison to our AP1 mutants. Then our second question, can the mutant AP1 protein substitute for AP1 function? And the answer is yes, it definitely can. It has petals, it has 1 flower per pedicel, and it looks very much like our positive control and like wild type. So what does all of that mean? So the farnesylation motif in the AP1 protein is not necessary for proper AP1 function, because the mutant AP1 protein that we mutated, where the farnesylation doesn't happen, can rescue these AP1 mutants. So that shows that it's not really doing anything very necessary for protein function. And it looks like the functional divergence between AP1 and full has occurred due to changes both in regulation and in the coding sequence, because full can only partially rescue AP1 mutants when it's expressed in the AP1 domain. So when we put it in the same place where AP1 normally is, it can partially substitute for it, but it still can't make petals. So something in the coding sequence is still different and changing the function. And our work on the consequences of gene duplication will shed light on how gene duplications affect floral morphology and how we see that in all the different